So DNA is a double-stranded helix, but how do we know that, right? So that journey actually happened about mid-century. So it involves Watson and Crick, and they were studying different types of structures using a technique called X-ray crystallography, which was being used by a few labs at the time. So they were having a really hard time understanding, or predicting the structure. And like many other labs, they were working, relatively hard on it. However, they went to this one lab, and they visited Maurice Wilkins. He was over at King's College in London. And Rosalind Franklin was one of his colleagues, which apparently he thought was his underling, but was not. And he shared her data, including a famous X-ray image of DNA that she had produced. And as the current story goes, I guess what's said is Watson and Crick ran back to their lab and took the data and published it, faster than Rosalind did. And as such, were considered to be the founders of the structure. So here's Watson and Crick looking at a structure that Rosalind Franklin is believed to be primarily responsible for generating the first data to lead to the knowledge of this structure. This is Rosalind Franklin, and this is the famous photograph using X-ray crystallography, right? So you can kind of see down here the X point. And so this is showing kind of the rotation of the helical model, okay? So after they saw these images and looked at some of the data, they realized that this is wrapped into a double helix. They also determined, furthermore, that the sugar phosphate backbone is something that's on the outside, and that we have nitrogenous bases, and they're going to pair A to T and G to C, okay? Here's that twist, right? So think of it as like a ladder, and you give it a slight twist, and then you could almost imagine looking down on it and seeing this kind of faded image, right? So eventually what happened is, before Rosalind Franklin understood the significance of her work, and possibly was writing on a synthesis of it, which was very difficult for her to do, being a woman in mid-century England in science. What happened was this crack team of folks who checked out her data essentially put it together, published it before her, and got it out to the scientific community and took credit. So they didn't acknowledge her contribution in any way. What was interesting is that she, I guess, had actually written them a congratulations letter saying, like, wow, your results are uncannily similar to what I've gotten, and so just kudos to you, which was really interesting. So she, however, died from cancer in 1958, likely because this X-ray crystallography was using radiation, and so some of the protection techniques were not as well known. So, that's a possibility of why she develops cancer so early, some folks have said. And so she died in 1958, unknowing of what her contribution actually was. In 1962, Wilkins, Watson, and Crick received the Nobel Prize. And the Nobel Prize was never awarded after death, right? Because it'd be like Galileo every year over and over. But anyway, or Newton, or, so many others. Anyway, so that's one of the reasons some folks are starting to call it the Franklin-Watson-Crick base pairing model. So sometimes you do see it referred to in older literature as just simply the Watson-Crick model. So that gave us kind of a platform to start identifying genes, chromosomes, and understand eventually, of course, base pairing and how that works together to result in the storage and transfer of genetic information within the chromosomes of organisms. Right. Okay. I hope that was informative. Have a great evening.